Besides, the Ministry of Education and Training, Ministry of Agriculture and Rural Development, Institute of Agricultural Science in Vietnam, NGO Xuân Nguyen. The investigation on selection, complementary, protocol in the cultivation of oyster mushroom, Pleurotus sp and big cup mushroom, Pleurotus sp. Summary, agricultural doctoral, dissertation, Hanoi, 2018. 2. Research results are published at Vietnam Academy of Agricultural Sciences, Scientific Advisors. First Prof. Dr. Habel, Trin Tem Kiet. Second Asok, Prof. Dr. Dung Chung Luong. One still reviewer, Asok. Prof. Dr. Duong Van Hop, Duan reviewer, Asok. Prof. Dr. Ngo Din Bin. 3-yard reviewer, Asok. Prof. Dr. Trin Kuk Quang. Dissertation will be presented under appraisement of the scientific Assembly from Vietnam, Academy of Agricultural Sciences at 8:30 a.m., January 18, 2018. Dissertation could be found at First National Library of Vietnam, Second Library of Vietnam Academy of Agricultural Sciences. 1. Introduction. First rationale. Fungi has a crucial role in human life, economic society, as well as science research. Oyster mushroom, Pleurotus spp., was recognized as an important mushroom genus in terms of economy in fungi. In Vietnam, oyster mushroom is one of 5 mushroom species and commercial developed with the yield above 60,000 ton year. Report of the Department of Crop Production at Mushroom Development in North Vietnam Conference, Du Sun, Haiphong 22-9-2011. Big cup mushroom, Clitocybe maxima, contains various acid amine and mineral, molybdenum, cobalt, zan, with several health benefits. The content of protein in big cup mushroom is 30%. Presently, cultivation of mushroom in Vietnam have to face several challenges such as cultivation technology, spawn production, new mushroom selection. The results of this research, the investigation on selection, complementary protocol in the cultivation of oyster mushroom, Pleurotus sp, and big cup mushroom, Clitocybe sp, has a contribution to the development of cultivation of mushroom in Vietnam. Second, the aim of research. The aim of this research is to hybrid and select new strain mushroom with high yield, good quality traits, as well as investigate on protocol for spawn production, cultivation technology. Third, the content of research. 3.1. Collect, hybrid, and select new oyster strain. 3.2. Collect, select imported big cup mushroom. 3.3. Evaluation of genetic differences and identification of hybrid oyster mushrooms, imported oyster mushrooms, and big cup mushrooms by RAPD PCR marker. 3.4. Research on the technology of propagation of the first culture, intermediate, secondary culture of potential hybrid oyster and big cup mushroom. 3.5. Optimization of cultivation technology of potential pink. 7. Hybrid oyster mushroom strain, BI big cup mushroom strain. Fourth, the scale of research. 4.1. Oyster mushroom. Collect, estimate gene source of domestic and imported oyster mushroom strains. Using commercial imported mushroom as material for hybriding. Study on some biological traits of some hybrid mushroom strains. Evaluate. Select some potent oyster mushroom strains by using cultivation method. Evaluation of genetic differences and identification of hybrid oyster mushroom, imported oyster mushroom by using RAPD, PCR. Study on the technological condition for submerged fermenter culture and subculture of potential hybrid mushroom strains. Optimization of the technical condition for potential hybrid mushroom cultivation by using complex substrates. 4.2. Big cup mushroom. Qualitative, estimate gene source of imported big cup mushroom strains. Research biological characterizations of imported big cup mushroom strains. Evaluation of genetic differences and identification of some big cup mushroom strains by using RAPD, PCR. 2. Study on the technological condition for submerged fermenter culture and subculture of some potential big cup mushroom strains. Optimization of the technical condition for potential strain cultivation by using complex substrates. 5th, contributions to scientific sense and practical sense. 5.1. Scientific contribution. The results of this research provide a database of biological traits as well as required conditions for optimization. The growth of hybrid oyster mushroom and big cup mushroom strains. These results can be considered as a material source for further studying on propagation technologies, cultivation on liquid culture medium and soft substrates. According to achieved results, there are some differences in terms of genetic of imported, domestics, and hybrid strains. These differences have a correlation with morphology and biological traits of researched strains. The results of this study can be used as a fruitfully reference in the further research regarding to development of mushroom cultivation technology in Vietnam. 5.2. Practical contribution. The results of this study achieved 1 hybrid oyster mushroom and 1 big cup mushroom strains, which have good quality in terms of yield, nutrient, and could be suitable for cultivation in Vietnam. This study successfully completed technological condition for submerged fermenter culture, cultivation for potential strains. This protocol have a good application in the cultivation of mushroom with industry scale. These results will diversify the types of cultivated mushrooms and promote the development of mushroom cultivation in our country. 5.3. The new finding of the research. The present study is highly logical from collection gene source, hybridization, screening to basic traits for potential mushroom strains. Conducted successfully protocol for producing spawn. Cultivate hybrid mushroom, big cup mushroom by using submerged fermenter culture and complex substrates with several advances, high yield, economic efficiency. The preliminary investigation on hybrid mushroom with mating of monokaryotic mycelium by hyphal fusion method in Vietnam. Chapter 1. Literature Review. Point 1. The role of oyster mushroom and big cup mushroom. Oyster mushroom, big cup mushroom contain a huge nutritional and pharmacy value with high flavor. There are several researches on oyster mushroom and big cup mushroom in the world. According to these researches, oyster mushroom, big cup mushroom can be used as a useful material in the treatment of cancer, HIV virus, as well as enhance immunity. Besides, cultivation of oyster mushroom and big cup mushroom contribute to protect environment. 2. General introduction to oyster mushroom and big cup mushroom. Oyster mushroom belong genus Pleurotus faricum. 1871. Scientific name? Pleurotus spp. Genus Pleurotus have several species with various at color, shape. This species has a high nutritional and pharmacy value. 3. Big cup mushroom, scientific name, Clitocybe maxima, P. Garten, G. May. And Sherb, P. Fear. Pilsk. Zerbst, 1231871. According to Kirk et al., 2008. Clitocybe genus contain about 300 species with widely distribution, especially north temperate. Big cup mushroom with English name giant Clitocybe. This species have a high nutritional and pharmaceutical value. Point 3. Influence of some factors on the growth of mycelium and fruit body. Oyster mushroom, big cup mushroom. There are several factors that influence on the growth and development of oyster mushroom and big cup mushroom. These factors can be divided into 3 typical factors. Outside conditions such as temperature, humidity, light, pH, CO, 2 concentration and wind speed, substrate, microorganism in ecology system. 1.4. The research and production of mushrooms in the world and Vietnam. After studying overview of researches in mushroom production in the world and Vietnam, the author found that there were many varieties of hybrid mushroom strains such as oyster mushrooms, button mushroom, shiitake mushroom. These strains can be used as useful materials for cultivation in industry scale by using liquid spawn and complex substrate. In generally, Vietnam has had many researches on edible mushrooms and medicinal mushrooms. However, these researches focus on only classification, selection, and building technology of spawn production, cultivation, preliminary processing, and preservation of mushrooms. But in Vietnam, there are very few research on select new hybrid strain, especially at the time of writing. No study on new hybrid oyster mushroom selection by using breeding method was published research. In addition, the research on the selection and construction of propagation and breeding techniques of edible mushrooms on complex substrates by using liquid spawn is still limited. Most of the mushroom strains such as oyster mushroom, big cup mushroom were still cultivated on traditional materials with the solid culture. The topic of this research is to explore new technologies, researching, selecting, and completing techniques for cultivating some new edible mushrooms, and contributes to improve the scientific and practical basis in the field of mushrooms cultivation technology in Vietnam. Besides, the results of this research also contribute to diversify types of cultivated mushrooms, meet the requirement of consumers, and promote the development of mushroom production in Vietnam. Chapter 2. Material and Method. 2.1 Material. 2.1.1. Spawn mushroom. Spawn mushroom included 9 oyster mushroom spawn, 4 big cup mushroom strains. Most of these strains were collected from overseas from 2005 to 2012 strain plus. 1 was officially recognized and cultured in Vietnam. 2.12 Reagents. Reagents were used for DNA isolation. 20 RAPD primers were used for different genetic detection. Reagents were used to produce pure culture and subculture. Materials were used to produce commercial spawn. 2.1.3 Conditions. Equipment used in experiments. 2.2 Method. 4. Methods was used, including the method of studying biological characteristics according to Trin Tom Kiet, 2012. The method of cultivation according to Din Xuan Lin et al., 2012. Hybridization method. Techniques for the extraction of the haploid spores are carried out according to Mohammadi E. Goltepe, 2007, Chaba Hajdu, 2008. Determination of pair coupling ability of single spore isolates by two-point grafting technique, Kotis Thani, 2003. Big cup mushroom, Clitocybe maxima. Cultivation technique was conducted according to Kuo, 2008. Protocol cultivation of hybrid oyster mushroom, Din Xuan Lin et al., Hai Ngin Mui Hai. The DNA extraction procedure is performed according to Alam et al., 2009. Research method in liquid spawn period, Yan et al., 2003. Nguyen Thi Beach. Tui Hai Ngin Mui Bon. 2.2.1 Collection, breeding, and selection of new oyster mushrooms. Experiment 1. Evaluation of imported oyster mushroom strains and domestic commercial strains. Control. Experiment 2. Hybridization of some commercially potential oyster mushroom strain that have been cultured in Vietnam. Experiment 3. Research on the biological traits of hybrid oyster strains. Experiment 4. Evaluation, selection of some potential strains in cultivation. 2.2.2 Collection and selection of imported big cup mushroom. Experiment 5. Collection and evaluation gene source of imported big cup mushroom. Experiment 6. Research on the biological characteristics of imported big cup mushroom. 2.2.3 The RAPD-PCR method combined with morphological traits to identify hybrid oyster mushrooms, imported oyster mushrooms, and big cup mushrooms. 2.2.4 Research on the technology of propagation of the first culture, intermediate. Secondary culture of hybrid oyster strain and big cup mushroom strain with high potential. Experiment 7. Influence of the submerged condition on the growth of the hybrid mushroom and big cup mushroom. Experiment 8. Effect of shaking regime on the growth of the hybrid mushroom and big cup mushroom in submerged medium. Experiment 9. Effect of incubation. Time on the growth of the hybrid mushroom and big cup mushroom. 2.2.4.2 Study on the technological condition for submerged fermenter culture of hybrid oyster mushroom and big cup mushroom in intermediate phase, second. Experiment 10. Influence of ingredient medium on the growth of hybrid oyster mushroom, big cup mushroom in intermediate phase. Experiment 11. Influence of oxygen concentration on the growth of hybrid oyster mushroom, big cup mushroom in intermediate phase. Experiment 12. Effect of spawn rate on the growth of hybrid oyster mushroom, big cup mushroom in intermediate phase. Experiment 13. Effect of incubation. Time on the growth of hybrid oyster mushroom, big cup mushroom in intermediate phase. 5. 2.25. Optimization of potential P7 hybrid oyster mushroom strain, BI big cup mushroom strain, cultivation technology. 2.2.5.1. Optimization of the technical condition for potential hybrid oyster mushroom, big cup mushroom cultivation on complex substrate by using seed spawn, traditional technology. Experiment 14. Study on the effect of composting method, medium formula on the growth, development, and yield of big cup mushroom, oyster mushroom. Experiment 15. Determine the optimal sterilization time. Experiment 16. Influence of medium formulations on fiber growth, fruit development, and yield of P7 hybrid mushroom and BI big cup mushroom. Experiment 17. Effect of liquid spawn age on the growth, development, and yield of hybrid mushroom and big cup mushroom. Experiment 18. Influence of the rate of original spawn on the growth, development, and yield of big cup mushroom, oyster mushroom. 2.3. Method of preparation of medium and experimental conditions. 2.4. Method of experimental layout. The experiment was arranged 3 times repeated and conducted as follows. OS Grade 1. 6 triangular vases, 500 milliliters, formula on 1 replication, each containing a duoratus milliliter triangular medium. Total 18 triangular vases, formula 3 repetitions. Intermediate, 3 Duran 5,000 milliliters, formula 1 repetition, each Duran contains 3,000 milliliters medium. Total 9 Duran cylinders, recipe 3 repetitions. Plus in the culture period, each formula is 60 bags of raw material, 1.4 kilograms, bag. Total 180 raw materials, experimental formulas, 3 repetition. Monitor the growth rate of 30 bags on test formula. Take the sample by 5 points of the diagonal. Each point is 6 bags. Traits. Monitor growth characteristics in pure cultures. Monitor growth characteristics in the juvenile phase. Plus KLC. Pellet size, M. Fiber biomass, G or 1,000 milliliters. Plus KLC density. Monitor growth characteristics during the culture period. 2.5. Data processing methods. Results of the study were processed by biological statistical method using Excel software and Iristat 5.0 software. 2.6. Place and time of experiment. Experiments were conducted mainly at the Center for Mushroom Research and Development, Agricultural Genetics Institute, Institute of Microbiology and Biotechnology, National University of Hanoi, Van Giang, Hung Yen Mushroom Center, and Vietnam National University of Agriculture. Experiment period, from 11 divided by 2012 to 6 divided by 2016. Chapter 3. Results and discussion. 3.1. Collection, breeding, and selection of new oyster strain. 3.1.1. Collect and evaluate gene source of imported and domestic commercial oyster. 6. Commercial oyster mushroom strains that were collected in Vietnam or imported from other countries during the period 2005-2010 have been evaluated through cultivation. The results are shown in Table 3.1. Table 3.1. Characterization of mycelium morphology, fruiting body and yield morphology of the commercial imported oyster mushroom strains. STT strain source, mycelium fruiting body. Average yield %. 1. Pl. 1. USA is being preserved in Vietnam. Mycelium is white ivory uniform high density. In mature phase, mycelium has the formation of the yarn braid. In the old phase, mycelium is white yellow. Mycelium growth rate 8.5 plus or minus 1 m a day. Fruiting body is white ivory, trumpet-shaped 3-7 cm diameter mushroom capsule, clustered mushroom, 5-12 fruiting body a cluster. Sweet and fragrant, grows well at 24-32. 0. C. Fig 3.1 National Oyster Mushroom Strain Pl. 1. 70 plus 5. 2 p. 11. Fao is being preserved in Vietnam. Mycelium is white, density is distributed evenly. In the mature phase, mycelium is dark white. In the old phase, mycelium is yellow. Mycelium growth rate 8.5 plus or minus 1.2 m day. Fruiting body is light purple. 8-15 fruiting body cluster with cap diameter is 3-7 cm, thick, sweet, and fragrant. Fig 3.2, strain P. 11. 60 plus 5. 3 p. 12. China is being preserved in Vietnam. Mycelium is white uniform high density. In mature phase, mycelium is ivory with high density. In the old phase, mycelium is white yellow. Mycelium growth rate 7.50.7 m day. Tukdo muksoi. Baizochum numskom dochum bayamangay. Fruiting body is purple, dish-shaped, 4-9 cm diameter cap, 3-5 fruiting body cluster, sweet and fragrant, grow well at 12-22. 0. C. Fig 3.3 strain P. 12. 50 plus 5. 4 SDL. Taiwan is being preserved in Vietnam. Mycelium is white, density is distributed evenly. In the mature phase, mycelium is dark white. In the old phase, mycelium is yellow. Mycelium growth rate 7.5 plus 1 m day. Fruiting body is gray, 3-7 cm diameter cap, sweet and fragrant, grow well at 15. 24. 0. C. Fig 3.4 strain STL. 45 plus 5. 7. 5 CP. Lao is preserved in Vietnam. Mycelium is white, density is distributed evenly. In the mature phase, mycelium is dark white. In the old phase, mycelium is yellow. Mycelium growth rate 9 plus 0.6 m a day. Fruiting body is gray, sweet, and fragrant. Grow well at 18 minus 28. 0. C. Figba do chum nam chung nam sa, sepia. 67 3. 3.1.2. The result of hybrids oyster mushroom from imported commercial mushroom that have superior traits and have been cultivated in Vietnam. 3.1.2.1. Create haploid lines. By using single-spore isolates, the major haploid lines of the two strains were made from PL1 and CP. The total number of single lines is 146 lines. In addition, haploid lines of three other strains, P11, P12, SDL, were conducted. The total number of single lines from the 5 strains was 220 lines. Table 3.2. 3.1.2.2. Hybrid mycelium from haploid lines. Fig 3.6. Types of morphology of mating site of haploid line. The evaluation was carried out by observing the mycelium under the microscope. Through evaluation, we selected 18 of the total of 225 pairs of hybrids. The list of generated hybrids is shown in Table 3.3. Table 3.3. The list of generated hybrid strains. No. Hybrid strain. No. Hybrid strain. No. Hybrid strain. No. Hybrid strain. 1 ampere 6, XB9, 5 amperes 2, XB3, 9 amperes 7, XB8, 13 amperes, XB8, 2 amperes 3 XB3, 6 amperes 10 XB10, 10 amperes 1 XB3, 14 amperes 1 XB1, 3 amperes 10 XB11, 7 amperes 11 XB10, 11 amperes XB6, 15 amperes 2 XB2, 4 amperes 11 XB11, 8 amperes 7 XB7, 12 amperes XB5, 16 amperes 4 XB4, 17 amperes to XB15, 18 amperes on XB12. The generated hybrid strains are preserved under pure conditions and conducted as well as fruiting body formation studies in the laboratory. Based on the results of the evaluation of the crosses created in the pure environment, 7 hybrid strains have been selected to evaluate the biological characteristics for the further experiments. 3.1.3. Results of the study on biological characteristics of hybrid oyster mushroom strain. We selected 7 hybrids with outstanding characteristics to evaluate biological characteristics in further experiments. PL1 strain was used as a control strain. 3.1.31. Evaluate the growth and development of hybrid oyster mushroom strain in the mycelial phase. 8. Table 3.4. The growth and development of hybrid oyster mushroom strain in the mycelial phase. Parameter. Strain. Mycelium growth rate, m. Characteristics of mycelium. A6, XB9. P1, 0.5. Mycelium is white, strong, and evenly distribution. A3XB3. P2, 541. Mycelium is white, grow well, and callus formation on surface cultivated bag. A10XB11. P3, 416. Mycelium is white, strong, and evenly distribution. A11, XB11, P4, 208. Mycelium is not clear, weak. A2, XB3, P5, 333. Mycelium is white, strong. A10, XB10, P6, 375. Mycelium is white, strong. A11, XB10, P7, 416. Mycelium is white, strong. PL1, 0.5. Mycelium is white, strong. LSD, 0.05, 0.26. Sieber-Cent 3.7. Table 3.4 illustrated the growth rate of the mycelium and mycelial density were quite different among hybrid oyster mushroom strains. 3.1.3.2. Morphology. Characterization of hybrid oyster mushroom. Table 3.5. Morphology characterization of hybrid oyster mushroom. Time for fruiting body formation. Days. Morphology characterization of fruiting body color. Fruiting boy cluster. Diameter cap, cm. Diameter stipe, Length stipe is to P1. 31 2 dark purple 265 3004 0A4 4.7. P. 2. Fruiting body formation was not occurred. P. 3. 31s2 dark purple, 355, 3 of 5 or 5, 6.1. P. 4. Fruiting body formation was not occurred. P. 5. 42 plus 3 dark purple, 1746, 03, 4.5. P. 6. Fruiting body formation was not occurred. P. 7. 30 plus 1 dark white, 96, 3, 402, 3.5. PL. 1. 32 us 2 white, 83, 303, 3.4. LSD. 0.05. O45. Mach 0.09. 0.22. C%. 4.99. 9.2. 14 epsilon 8. 2.7. 3.1.3.3. Yield of hybrid oyster mushroom strain. Table 3.6. Yield of some hybrid oyster mushroom strains. Dry substrate weight, K. Moist substrate weight, K. Fresh mushroom weight, K. Efficiency of fresh mushroom dry substrate weight, %. P. 1. 36,384. 889, 24.5. P. 3. 36,384, 1129. 31.1. P. 5. 36,384. 265. 7.3. P. 7. 36.3. 84, 1848, 50.6. PL. 1. 36.3. 84, 20, 58. 56.7. LSD. 0.05, 3.02. C% 5.4. 9. Table 3.5, Table 3.6 showed that the color morphological characteristics of hybrid oyster mushroom strains were very different. Specifically, strain P2, P4, P6 had a short time of mycelial phase, but could not form fruit. In constat, yield of P7 strain was 50.6%, approximately same to the PL1 control strain, yield with 56.7%. For hybrid strain, the first generation is often stressed, and the yield is lower than that of their parent. This statement also agreed by Mohammadi Goltepe et al., 2007, when studying oyster mushroom Pleurotus austriatus. 3.1.34. Evaluation and selection of some potential mushroom strains in cultivation. Selection of commercially potential oyster mushrooms and hybrid mushroom strains were carried out. The sources of materials are shown in Table 3.7. Evaluate the growth rate, development of mycelium of test strains. Table 3.8. Growth rate, development of mycelium oyster mushroom strain on cultured media. Unit, day before inoculation. Strain. Mycelial phase. Day. The growth rate of mycelium M average time for the fully growth of mycelium on surface bag, days. 14D, 16D, 18D, 20D, 22D. P. 1. 1143, 558, 65, 7, 25s, 2. P. 7. 736, 758, 7387, 81, 16, 5, 22, plu 1. P. 8. 7. 36, 353. 5606, 482, 11522 plus 1. P. 9. 837537586, 114232. P. 10. 726, 739, 5527, 487, 27 plus 2. P. 11. 9. 35, 263, 38204, 113, 24s 2. P. 12. 934486181 8106 25 plus 2. P. 13. 7415 0679 112022 plus 2. PL. 1. 737 357 2769 011523 plus 1. CP 7365776 889 113 23 plus 1. And the percentage of contamination and morphology of mycelium on substrate, table. 3.9. 10 strains are capable of well growth and development. Mycelium on surface substrate is white and very thick. In the P7 strain and the P8 strain, the density of mycelium is thicker. The color of the surface substrate is more uniform than the rest of the formula. The contamination rate of the experimental formulations is, of which oyster mushroom strain P7 has a very low prevalence of 3.57%. This result explained its disease resistance better than the control, PL1. Evaluation of formation and development of feuting, Bobby of oyster mushroom on waste cotton substratum, Table 3.10, showed that the time for fruiting body formation after inoculation were about 40 days with no significant differences among strains. Each strain has its characteristic fruiting shape, particularly the size of mushroom cluster of P7 strain is large, thick, and uniform. The number of mushroom cap on each cluster is not excessive. The uniformity is an essential element for high yield potential. The fruiting body of strain P1 was purple, relatively good, short and thick stipe, thick mushroom wing with number of 3-5 wings each mushroom cluster. These features are suitable for the purpose of mushroom breeding. Harvest productivity of oyster mushrooms of 3 trials. 10. Table 3.11 Yield of hybrid and imported oyster mushroom. Strain average yield cage bag. Average yield percent. P1 0.13, 25.29. P7 4.43, 83.66. P8 Fluoro 1733.07. P9 Arno 9 17.51. P10 0.14, 27.24. P11 0.14, 27.24. P12 0.01, 1.95. PL1 0.38, 73.93. CP 0.37, 71.98. LSD 0.05, 3.09. CV percent 6.25. Table 312. Quality analysis of P7 hybrid oyster mushroom strain and PL1 control strain. The A results were analyzed at the National Center for Food Analysis and Inspection, Institute of Food Technology. No. Nutrient unit. Results. Test method. P7 strain PL. 1 strain. 1 protein content G 100 grams. 205. 1.78. TCVN 8,125 2009. 2 fat content 100 grams. 430.12. TCVN 8,136 2009. 3. Carbon hydrate content G 100 grams. 2.54. TCPTN 1 HPLC. 4 vitamin C content M 100 grams. 351. 0.78. JAS SUP 60. 5 vitamin B1 content M 100 grams. O20. 0.12. JAS soap 57. 6. Canxi K content M 100 grams. 26.5 28.0. TCVN 1,526. 1. 2007. 7. Phospho content G 100 grams. 14 to 15. 14.24. TCVN 6,271 2007. 8. Iron fog content 100 grams. 153. 1.36. AOAC 999.11. Nuff moist percent. 88 and 66. 86.54. TCVN 4069 2009. Based on the results of the analysis, P7 hybrid mushroom strain was fully converged with higher yield characteristics than the controls of 10%, Table 3.11. The quality of fresh mushrooms was higher than that of control, Table 3.12. Moreover, P7 strain can widely adapt to temperature and disease resistance. The P7 hybrid mushroom strain has been selected to carry out further experiments on the technology of spawn production and cultivation. 3.2. Collection and selection of imported big cup mushroom strains. 3.2.1. Collection, evaluation of gene source of imported big cup mushroom, Clitocybe maxima, GAN10X link. Big cup mushroom strain was imported from China and Taiwan in 2005, 2006, and cultivated by using complex substrate, QOM, 2008 April. Results showed in the table 3.13, 3.14, and 3.15. 11. Table 3.15. Characteristics of growth, development of big cup mushroom strains. Strain. Mycelium formation. Day. Mycelial phase. Day. Color. Stipe length, C. Cap diameter. cm. BI. 33 plus 2. 56 plus 2 dark brown when young phase, many white scales, light brown when mature phase. Fig 3.7 strain, BI, La, 23-1, 13.6. BI, 0. 36 plus 2, 59 plus 2 dark brown. When young phase, many white scales, light brown when mature phase. Fig 3.8 strain, B Sol, 0. 23-4, 13.2. BI, 1. 39 plus 2, 62 plus 2 dark brown. When young phase, very few white scales, light brown when mature phase. Fig 3.9 strain, BI, 1. 18-5, 10.8. BI, 2. 37 to 59, 2 dark brown. When young phase, many white scales, light brown when mature phase. Fig 310 strain, BI, 2. 18-1, 11.6. LSD, 0.05. 081, 0.38. CV percent, 201, 1.6. Notices. Time for mycelium formation, day. From inoculation to mycelium growth fully on surface substrate. Mycelial phase, day. From inoculation to first fruiting body harvest. Compositional characteristics of yield of big cup mushrooms, Table 3.14. Based on results from Table 3.13, Table 3.14, Table 3.15, together with observations during the empirical study, the results showed that BI0 strain belonged superior advantages in term of stem length, stipe diameter, fruiting body weight. Beside BI, a big cup mushroom strain, BI0 was evoluted by using sensory method, focusing on the beauty, better taste, more preferable than the breeded BI1 and BI2. From the results of our research, author chose the strain with strong vigor BI0 as a material to conduct in further study of some biological traits. 12. Table 3.15. Evaluation of yield and quality of big cup mushrooms. Strain. Yield. Percent. Storage time. Days. Sensory evaluation. BI. 49-8-10. Stipe and cap are sweet, crispy, typical fragrance. Fresh for long cryopreserved time. Easy to pack and less broken cap. BI. 0. 48.3-10 sweet, crispy, typical fragrance. Fresh for long cryopreserved time. Easy to pack, less broken cap. BI. 1. 40-2-8. Hard, dry, fresh for long cryopreserved time. Easy to pack, less broken cap. BI. 2. 37-6-8. Sturdy, fresh for long cryopreserved time. Cap break easily during transportation. LSD. 0.05. 4.65. CV percent 5.6. 3.2.2. Some biological characteristics of imported big cup mushroom, BI. What about? 3.2.21. Influence of nutritious supplement on the growth of mycelium. Influence of glucose concentration on mycelium. Growth of big cup mushroom. Fig 3.11. Influence of glucose. Concentration on mycelium. Growth of big cup mushroom. Influence of peptone concentration on mycelium. Growth of big cup mushroom. Fig 3.12. Influence of glucose. Concentration on mycelium. Growth of big cup mushroom. Influence of KH. 2. PO. 4. Concentration on mycelium. Growth of big cup mushroom. Fig 3.16. Influence of KH. 2. PO. 4. Concentration on mycelium. Growth of big cup mushroom. Strain. Concentration KH. 2. PO. 4. 1,000 milliliters. R.5112. 2.5. BI. 0.41 plus 0.01. 0.43 Daros 0.2. 0.47 0.02. 0.49 plus 0.01. 0.47 plus 0.01. 0.46 plus 0.01. BI. 0. 0.38001. 0.4002. 0.45 0.02. 0.48 0.01. 0.46 plus 0.01. 0.44 0.01. 13. Influence of pH on mycelium growth of big cup mushroom. Fig 313. Influence of pH on mycelium growth of big cup mushroom. Influence of temperature on mycelium growth of big cup mushroom. Fig 3.18. Influence of temperature on mycelium growth of big cup mushroom. Temperature. Parameter. 20-22. 0. C24-26. 0. C28-30. 0. C. The growth rate. M and Gay. BI. 5.101, 6.201, 6.8 or 0.1. BI. 0. 4.7 0.1, 6.0 plus 0.1, 6.5 plus 0.1 characteristics of mycelium. BI. Mycelium is light white, high density. Fruiting body formation is early when mycelium do not growth fully solid medium. Mycelium is dark white, fruiting body can be occurred. Mycelium is white with height density, growth rate is quick, fruiting body can be occurred but lately. BI. 0. Mycelium is thin. Fruiting body formation occurred when mycelium do not growth fully solid medium. Mycelium is dark white. Fruiting body can be occurred but lately. Mycelium is thin. Fruiting body can be occurred but lately. From the results of the collection, evaluation of biological characteristics, this research have been selected BI big cup mushroom strain with full convergence of precious characteristics of high yield, beautiful fruiting body shape, great quality, broad adaptability to temperature conditions and disease resistance as a material for further experiments. 3.3. Evaluation of genetic differences and identification of hybrid oyster, imported oyster, and big cup mushrooms by RAPD PCR marker. 3.31. Evaluation of genetic differences and identification of hybrid oyster and imported oyster mushrooms by RAPD PCR marker. 3.31.1. Identification of hybrid oyster and imported oyster mushrooms by RAPD. PCR marker. The use of RAPD primers combined with morphological characteristics can be accurately identified the mushroom strains. OPA 2 primer. Identify exactly sample number 6. OPA 18 primer. Identify exactly sample number 2. 14. S208 primer. Can precisely identify sample number 1 and 4. S266 primer. Identify exactly sample number 1 and 8. S205 primer. Identify exactly sample number 4 and 5. S300 primer. Identify exactly sample number 2, 3, and 7. Furthermore, from the result of gel electrophoresis with primers, S300, S285, S208, OP18, and OPN5, we achieved evidences of the relationship between parents, pair 5, 6, and hybrid strain, pair 9, num 10. For S300 primers, if found band at any site of hybrid strain, the band was also found in the parent strain with same position. In contrast, if both parents appear band in the same position, 100% of the hybrids also have band in same position. Samples 6, parent samples, and samples 8, 9, hybrid strains, appeared in all the same positions in 4 primers, S285, S208, OPA18, and OPN5. Except for sample 5, parental sample, there are some different band compared with the other 3 samples. Fig 3.5, morphology of parent oyster mushroom and hybrid strain. Parent, sample 5, CP. X sample 6, pool 1. On hybrid strain, sample 9, P 7. And sample 10, P 1. 3.3.1.2. Evaluation of genetic differences of hybrid oyster and imported oyster mushrooms by RAPD PCR marker. Coefficient 0460530600067 074080 10 MW 1 4 5 6 9 10 8 2 3 7. Fig 3.15. Diagram of the genetic relationship among 10 studied mushroom species. 3.3.2. Evaluation of genetic differences and identification of imported big cup mushrooms by RAPD PCR marker. 3.3.2.1. Identification of imported big cup mushroom by molecular markers. The use of RAPD primers combined with morphological characteristics can be accurately identified the mushroom strain. OPO15, OPO18, SEN S239 primers. Identify exactly the sample number 4. OPA18, OPO18, S239, S300, S285 primers. Identify exactly sample number 3. S300 primers. Identify exactly sample number 2. OPA18 primers. Identify exactly sample number 1. 15. 3.3.2.2. Evaluation of genetic differences of imported big cup mushrooms by RAPD. PCR marker. Fig 3.16 diagram of the genetic relationship among 4 studied mushroom species. 3.4. Research on the technology of propagation of the first culture. Intermediate. Secondary culture of potential P. 7 hybrid oyster strain and BI big cup mushroom strain. 3.4.1. Research on the technology of propagation of the first culture of potential P. 7 hybrid oyster strain and BI big cup mushroom strain. 3.4.1.1. Influence of the submerged condition on the growth of the hybrid mushroom and big cup mushroom. Table 323. The growth of the hybrid mushroom and big cup mushroom in submerged medium. Strain formula. Pellet density. The size of pellet M. Pellet characteristics. Mycelium. Biomass. 1,000 milliliters. Big cup mushroom BI. La. I++. 1.8. Culture is clear. Pellet has short fringe. Smooth. 16.8. 2. Plus 0.9. Culture is clear. Pellet has short fringe. Smooth. 21.4. 3. Plus 0.95. Culture is clear. Fluid is suspension. 26.5. IV plus 1.2. Culture is clear. Fluid is suspension. 29.5. V plus 1.6. Pellet is suspension. 24.8. LSD 0.05. 0.24. 1.6. C%. 10:1. 3.7. Hybrid oyster strain. P. 7. La. I plus 1.67. Pellet is smooth. Dispersed liquid 21.8. 2. Plus 1.25. Pellet is smooth. Suspension 24.4. 3 plus 1.05. Pellet is smooth. Suspension 28.5. IV plus 0.88. Pellet is smooth suspension. 32.2. B plus 1.35. Pellet is smooth. Dispersed liquid 26.8. LSD 0.05. 0.25 2.0. CV% 1009 4.2. Notes. Pellet density, KL smil. Pellet density, KLC, plus KLC 1030 K 1 milliliter culture, Psychol 231, 60 K 1 milliliter culture, plus Psychol 61, 90 KLC, 1 milliliter culture, plus KLC 91, 120 K 1 milliliter culture, plus KLC 120 divided by 1 ml culture. This convention applies to the next statistic tables. Table 3.30 showed that hybrid strain grows best on the formula for 200 grams potato, plus 100 grams rice bran, plus 100 grams fresh mushroom, plus 0.5 grams MgSO 4.7, H2O, plus 0.5 grams KH2, PO4 plus 20 glucose. Coefficient 0. 76079 082085 08809 1 MW 1 2 3 4 BI 0 BI 1 BI 2 16. Big cup mushroom strain can grow best on formula for 100 grams bean sprouts plus 2.5 grams CNM plus 3.5 grams peptone plus 0.5 grams MgSO 4.7, H2O plus 1 gram KH2, PO4 plus 20 G glucose plus 1 mg vitamin B1. 3.4.1.2. Effective shaking regime on the growth of the hybrid and big cup mushrooms in submerged medium. Table 3.24. Effective shaking regime on the growth of the hybrid and big cup mushrooms in submerge, medium. Strain. Shaking regime. Round minutes. The size of pellet, M. Pellet density. Mycelium biomass. 1,000 milliliters. Big cup mushroom strain, B. 101.8 plus 24.6. 120. 1.4 plus 28.8. 140. 1.2 plus 31.9. 160. 1.1 plus 29.4. 180. 0.7 plus 24.2. LSD. 0.05. Oral 35 1.9. C percent 15.4 3.8. Hybrid oyster mushroom strain, P. 7. 101.68 plus 28.5. 120. 1.15 plus 37.8. 140. 0.8 2 plus 31.2. 160. 0.75 plus 30.4. 180. 0.7 2 plus 26.7. LSD. 0.05. 432.1. C percent 16.4 3.8. 3.4.1.3. Effective incubation. Time on the growth of the hybrid and big cup mushrooms. Table 3.25. Effective incubation time on the growth of the hybrid and big cup mushrooms. Strain. Parameter. Time. Airs. The size of pellet, M. Pellet density. Mycelium. Biomass. 1,000 milliliters. Pellet characteristics. BI. Big cup mushroom strain. 48 0.65 plus 11.2. Pellet is scattered, not attached to the bottle. 60 0.74 plus 18.6. Pellet is scattered, short fringe, smooth. Low density. 72 1.02 plus 26.2. Pellet combine tightly. Each other, short fringe, smooth, thick density, fluid suspension. 84 1.16 plus 29.6. Thick density, big size, short fringe, and smooth. 96 1.2 plus 31.9. Thick density, big size, short fringe, and smooth. 120 1.56 plus 31.4. Pellet reduced association. LSD. 0.05. Fluoro 21 1.52. C percent 11.23 3.7. P. 7. Hybrid oyster mushroom strain. 48 0.2 0 plus 13.4. Pellet is scattered, not attached to the bottle. 60 0.44 plus 28.8. Pellet is scattered, smooth, small. 72 0.68 plus 32.8. Pellet combine tightly each other, smooth, thick density, fluid suspension. 84 1.06 plus 38.2. Fluid suspension. Pellet is big and smooth. 96 1.15 plus 37.8. Pellet is big, smooth, and combine tightly each other. 120 1.33 plus 37.6. Unshaped. LSD. 0.05. Aura 15 1.5. CV percent 10.21 2.6. From the results obtained in Table 3.25, we found that mycelium biomass reached its maximum at 38.2 grams on 1,000 milliliters, with hybrid oyster mushroom and 31.9 grams, 1,000 milliliters, with big cup mushroom. 17. 3.4.2. Study on the technological condition for submerged fermenter culture of hybrid oyster and big cup mushrooms in intermediate phase second. 3.4.2.1. Influence of ingredient medium on the growth of hybrid oyster and big cup mushrooms in intermediate stage. Table 326. The growth of hybrid oyster and big cup mushrooms in submerged intermediate stage strain formula. Pellet density. Size of pellet, mem. Pellet characteristics. Mycelium. Biomass. 1,000 milliliters. BI. Big cup mushroom strain. I. plus 1.93. Culture is clear, pellet has fringe, smooth, density is low, 19.4. 2. Sauce 1.82. Culture is clear, pellet has fringe, density is middle, 27.8. 3. Sauce plus 1.78. Culture is clear, pellet has fringe, 33.9. Voi. Sauce plus 1.8. Culture is clear, pellet has fringe, 26.1. V. Sauce 1.96. Culture is clear, pellet has fringe, 22.4. LSD 0.05. 412.1. Severcent 4.1. 4.7. P. 7. Hybrid oyster mushroom strain. IPAWS 1.85. Culture is clear, pellet is smooth, 21.8. 2. Tlasa Sasvas 1.45. Culture is clear, pellet is smooth, 24.4. Treepsaw 1.25. Culture is clear, pellet solution is suspension, 28.5. IV plus sauce 0.95. Culture is clear, pellet solution is suspension, 31.5. Phos 1.3. 5. Culture is clear, pellet solution is suspension, 27.8. LSD 0.05. R2 2.3. Severcent 8.0. 4.7. The growth rate of big cup mushroom mycelium reached the highest on Formula 3, 2 grams high yeast, 2 grams peptone, plus 0.5 grams, MGSO 4.7, H2, O 15 grams glucose, 1.5 million thiamine, 1,000 milliliters culture. The optimum formula for the growth mycelium of hybrid oyster mushroom is Formula 4, CT4, 200 grams potatoes, 100 grams fresh shellfish, 100 grams rice bran, 0.5 grams. MGSO 4.7, H2O plus 0.5 grams, KIOPO4 plus 20 G glucose. 3.4.2.2. Influence of oxygen. Concentration on the growth of hybrid oyster and big cup mushrooms in intermediate stage. Table 3.27. Influence of oxygen. Concentration on the growth of hybrid oyster and big cup mushrooms in intermediate stage. Strain. Concentration of oxygen. VVM. Size of pellet. MKLC. Pellet density. Mycelium biomass. To 1,000 milliliters. BI. Big cup mushroom strain. 0.4. 51.7. 4 plus 18.6. Aura 55. 1.58 plus 23.2. Aura 65, 0.98 plus 32.8. Aura 75, 0.86 plus 29.8. Aura 85, 0.54 plus 24.6. LSD 0.05. 0.13, 1.9. CD% 622, 4.1. P. 7. Hybrid oyster mushroom strain. 0.4, 51.62 plus 20.7. Flora 55, 1.47 plus 28.9. Flora 65, 1.02 plus 29.8. Flora 75, 0.81 plus 31.6. Flora 85, 0.54 plus 28.8. LSD 0.05. Flora 97, 1.8. CV% 3.4, 3.5. 18. 3.4.2.3. Effective spawn rate on the growth of hybrid oyster and big cup mushrooms in intermediate stage. Table 3.28. Effective spawn rate on the growth of hybrid oyster mushroom, big cup mushroom in intermediate stage. Strain parameter. Spawn rate %. Size of pellet density. Mycelium biomass. 1,000 milliliters. BI. Big cup mushroom strain. 50.87 plus 24.8. 70.94 plus 30.2. 10. 0.95 plus 33.6. 12. 1.02 plus 31.1. 15. 1.24 plus 32.2. LSD 0.05. Doro 15 1.7. Severcent 8.33.1. P. 7. Hybrid oyster mushroom strain. 1.78 plus 20.6. 7 1.41 plus 22.5. 10 1.08 plus 32.8. 12 0.86 plus 28.4. 15 0.5 2 plus 28.4. LSD 0.05. 93 1.4. CV% 4.6. 2.9. 3.4.2.4. Effective incubation time on the growth of hybrid oyster and big cup mushrooms in intermediate stage. Table 329. Effective incubation time on the growth of hybrid oyster mushroom, big cup mushroom in intermediate stage. Strain parameter. Incubation. Time, Hrs. Pellet density. Mycelium biomass. G 1000 milliliters. Pellet characteristics. By big cup mushroom, M strain. 48 plus 21.2. Pellet is scattered, not attached to the bottle. 60 plus 24.6. Pellet is scattered, pellet has fringe, smooth, density is low. 72 plus 28.6. Pellet combine tightly each other, short fringe, smooth, thick density, fluid suspension. 84 plus 31.6. Thick density, big size, short fringe, and smooth. 96 plus 33.9. Thick density, big size, short fringe, and smooth. 120 plus 33.1. Pellet reduced association. LSD 0.05. 1.7. C% 3.6. P. 7. Hybrid oyster mushroom strain. 48 plus 14.3. Pellet is scattered. 60 plus 27.8. Pellet is scattered. 72 plus 33.8. Pellet combine tightly each other. Smooth, thick density, fluid suspension. 84 plus 37.8 fluid suspension. Pellet is big and smooth. 96 plus 36.8. Pellet is big, smooth, and combine tightly each other. 120 plus 36.6 unshaped. LSD 0.05. 1.55. C% 2.65. 19. 3.5. Optimization of potential P's of an hybrid oyster mushroom strain and BI, big cup mushroom strain cultivation, technology. 3.5.1. Optimization of the technical condition for potential hybrid oyster and big cup mushrooms, cultivation on complex substrate by using seed spawn, traditional technology. 3.5.1.1. Study on the effect of composting method, medium formula on the growth, development, and yield of big cup and oyster mushrooms. Results were shown in Table 330, 332, 3.33. Table 3.30. Effect of substrate composition, incubation method on the mycelium growth of big cup and hybrid oyster mushrooms. Strain incubation method. Formula. Time for mycelial growth, day. 50% bag, 100% bag. Fruiting body formation. By big cup mushroom strain. P. DC. CT1 21 36 43. CT2 22 43. CT3 18 31 37. CT4 DC 17 31 38. LSD 00519 2.3 2.7. C% 511 3.9 3.3. P. TN. CT1 19 32 39. CT2 2034 40. CT3 16 28 33. CT4 DC 15 27 32. LSD 0.0514 201 3.1. CV% 4.0 3.3 4.4. P. 7. Hybrid oyster mushroom strain. P. DC. CT1 17 29 43. CT2 13 21 28. CT3 15 24 31. CT4 DC 12 20 25. LSD 0.05 226 283.2. C% 86 6.4 5.7. P. TN. CT1 15 25 32. CT2 12 20 26. CT3 14 23 29. CT4 DC 12 9 25. LSD 0105 1.6 1.70. 2.3. C%, 6.0, 040, 4.7. The effective composting method, medium formula on the percentage of contamination, showed in Table 3.31. Table 332. The effect of composting method, medium formula on the yield of big cup and oyster mushrooms. Strain formula. Yield of first flushing, percent. Yield of second flushing, percent. Yield of third flushing, percent. Average yield, percent. By big cup mushroom strain. P. DC. CT1, 39.4, 42.8, 42.2, 41.5. CT2, 38.2, 41, 39.6, 39.8. CT3, 48.5, 52.7, 52.5, 51.2. CT4 DC, 48.4, 45.6, 48, 6, 47.2. 20. LSD05, 3.37. C% 4.0. P. TN. CT1 43.46, 45.3, 44.5. CT2 42.2, 41.8, 43, 42.5. CT3 55.6, 58.6, 57. CT4 control 49.2, 51 toto, 53.8, 51.4. LSDR 205 2.8. C% 3.1. P. 7. Hybrid oyster mushroom strain. P. DC. CT1 54 4 58 8, 63 56.5. CT2 78.2, 79.8, 77.5, 78.5. CT3 74, gate 666 68.5. CT4 control 78.2, 82.6, 81.4, 80.7. LSD 205, 3.7. C% 2.7. P. TN. CT1 75.6, 74 73.4, 74.6. CT2 92, 87 90, 89.5. CT3 80.6, 83 82.5, 82.3. CT4 control 81 to 83 2, 81 8, 82.1. LSDR 205 2.8. C% 1.6. Yield percent. Fresh fruit and body. Weight of dry substrate. X100. From the statistical results in Table 3.32 and the evaluation in Table 3.3 and Table 330, it was confirmed that the new composting incubation method had several advantages comparing with traditional method. With the new compost incubation method, the yield of hybrid oyster strain and big cup mushroom strain increased 14% and 11.3%, respectively. This result has great practical significance in the development of mushroom production. 3.6 to 1.2. Experiment 15. Determine the optimal sterilization time. Table 3.33. Effective sterilization. Time on the growth, development, contamination, and yield of big cup and oyster mushrooms. Formula. Sterilization time H. Contamination rate percent mycelial full growth on surface bag. Day. Fruiting body formation. Day. Average. Percent. Hybrid oyster mushroom. P. 7. CT2. 415212865. 64202688. 8612027882. 1095232877. LSD05. 18117 4.8. CV percent. 1009 3us 3.3. 3.4. Big cup mushroom. BI. CT3. 4253-24138I. 651-283-2582. 856-30-3357. 1010-23-24049. LSD05. 185-155. 246. 4.16. CV percent. 9.0, 3.0, 3.86. 4.4. 21. A note. CT2. 48% sawdust, 48% cottonseed plus 3% rice bran plus 1% KKO3. CT3, 39.5% sawdust plus 39% cottonseed plus 0.1% MnSO4 plus 0.1% KH2PO4 plus 8% corn power, 12% rice bran, 1.3% KCO3. A particularly interesting issue is yield. According to Table 340, the P7 hybrid oyster mushroom strain in the Formula 2 by using sterilization time for 6 ARs at 100. 0. C achieved the highest yield with 88.4 plus or minus 2, the yield of big cup mushroom B in the Formula 3 by using sterilization time of 6 HRs at 100. 0. C reached the highest 58.2 plus 2. 3.5.2 Optimization of cultivation. Technology of potential P7 hybrid oyster mushroom strain and BI big cup mushroom strain by using complex substrates with liquid spawn. Novel technology. 3.5.2.1 Influence of spawn source. Medium formulations on mycelial growth, fruiting body development and yield of hybrid mushroom P7 and big cup mushroom BI synthesis. Experiment 16. Results was given in Table 3.34 and 3.35. Table 3.34. Influence of spawn source. Medium formulations on mycelial growth, fruiting body development, and yield of hybrid oyster and big cup mushrooms. Strain. Formula. The growth rate. M-day. Mycelial density. Contamination rate percent. P7. Hybrid oyster mushroom strain. Liquid spawn. CT1 12.62 plus 8.3. CT2 16.75 plus 3.8. CT3 13.95 plus 6.4. CT4 DC 15.24 plus 4.2. LSD 0.05. 0.2 0.41. C% 4 3.8. Seed spawn. CT1 7.32 plus 7.6. CT2 10.6 4 plus 4.1. CT3 8.73 plus 6.3. CT4 DC 9.91 plus 4.4. LSD 0.05. 135 0.34. CV% 7.8 plus 3.2. BI Big cup mushroom strain. Liquid spawn. CT1 7.62 plus 6.3. CT2 8.45. SOS 7.8. CT3 9.96. SOS 5.4. CT4 DC 8.21 plus 6.2. LSD 0505403. CV% 3.4 2.5. Seed spawn. CT1 4.16 plus 8.2. CT2 5.52. LA 7.1. CT3 5.95. SOS 5.3. CT4 DC 5.78 plus 7.4. LSD 0503203. C% 3.2. 2.3. 22. Table 335. Influence of spawn source. Medium formulations on fruiting body development and yield of hybrid oyster and big cup mushrooms. Spawn. Formula growing mycelium covered the whole surface of the bag. Day. Fruiting body formation. Day. Average yield percent. P7. Hybrid oyster mushroom strain. Liquid spawn. CT1 18 58. CT2 1221 89.4. CT3 1324 81.5. CT4 control 1324 83.7. LSD 0.05 2.2 208 5.25. CV% 8.7 6.1 3.5. Seed spawn. CT1 2838 55.4. CT2 20 27 86.4. CT3 23 30 80.2. CT4 control 22 29 78.3. LSD 05 16265 6.1. CV% 307 5.1 3.4. BI Big cup mushroom strain. Liquid spawn. CT1 23 13 45.1. CT2 24 31 53.2. CT3 18 26 61.5. CT4 control 19 27 54. LSD 0.05 135 167 3.65. CV% 3.44 3.0 3.0. Seed spawn. CT1 32 4042.1, CT230 51.2, CT3 2833, 57.5, CT4 control 30 30s 49, LSD05 187 215 4.35, CV percent 3.3 3.4 4.7. Note: Formula CT1, CT2, CT3, CT control for each mushroom strain, Table 2.6, Table 2.7, were given in Appendix 1. The statistical results in Table 3.35 indicated that growing mycelium covered the whole surface of the bag. The fruiting body formation and harvest yield were significantly different among complex substrates. For P7 hybrid strain, the Formula 2 gave the highest yield with 89.4 plus or minus 2% by using liquid spawn and 86.4 plus or minus 2% by using seed spawn. While BI Big Cup mushroom strain, Formula 3 had the highest yield with 61.5 plus or minus 2% by using liquid spawn and reached 57.5 plus or minus 2% by using seed spawn. 3.5.2.2 Effective liquid spawn age on the growth, development, and yield of hybrid oyster and big cup mushrooms. Experiment 17. Based on the above results, the optimum of submerged spawn age of hybrid oyster mushroom strain was 84 HRs, while big cup mushroom was 96 HRs. These results is shown in Table 336. 23. Table 3.36. Effective liquid spawn age on the growth, development, and yield of hybrid oyster and big cup mushrooms. Strain spawn. Contamination rate, percent sol growing mycelium covered the whole surface of the bag, day. Fruiting body formation, day. Average yield, percent. P. 7. Hybrid oyster mushroom strain. Seed spawn, 6. 52134584. Liquid spawn, 48H16 819-32529, 60H8 6153057, 72H6 2142874, 84H4 5132 1875, 96H68 1529, 77.3, 120H10 4183451 shop, LSD 0.05, 1,087, 2 of 15, 3.85, C% 96, 573.24.3. BI. Big Cup Mushroom Strain. Seed Spawn. 11-2, 404656. Liquid Spawn. 48H15, 436, 46, 48 60H10, 433425668. 72-6, 6313857 soy. 84H6, 62835586. 96H6, 42733602. 120H11, 8313756 LSD 0.05, 2 by 218, 288, 3.25. C%, 12.8, 3.9, 4.41, 3.3. 3.5.2.3. Influence of the rate of original spawn on the growth, development, and yield of big cup and hybrid oyster mushroom. Experiment 18. Table 337. Influence of the rate of original spawn on the growth, development, and yield of big cup and hybrid oyster mushroom spawn. The volume of original spawn, MEL. Contamination rate, percent growing mycelium covered the whole surface of the bag, day. Fruiting body formation, day. Average yield, percent. P. 7. Hybrid oyster mushroom strain. 10-9, 5223-2655. 15-8-616 SAW. 25854. 20. 43122. 1905. 25491321. 896. 30. 5713208864. LSD 205. 168, 1205, 3.45. C% 14.2, 501, 500, 2.4. BI. Big Cup Mushroom Strain. 10-9-629. 37, 51.3. 15-608-23. 32, 53.6. 26, 719-29586. 25-5-817-27613. 30, 6217. 2757.5. LSD 005. 1 tone 92. 2 tone 25. 2 tone 3 is 3.26. Seabercent 14.7. 4 weight 42. 3.3. According to Table 337, it was determined that the most optimum rate of original spawn for the growth, development, and yield of big cup and hybrid oyster mushrooms was 25 milliliters bag and 20 milliliters bag, respectively. 24. Conclusions and recommendations. Conclusions. First, hybrid oyster mushroom P. 7. Strain can be adapted to the wide temperature range. Low susceptibility, high yield 89.5% big cup mushroom strain by can be adapted to wide temperature range. 22-30 L. C. Less disease, high yield with 61.5%, good quality, adaptable with several ecological regions in Vietnam. Second, the P. 7 hybrid oyster mushroom strain, BI, big cup mushrooms, other hybrid oyster mushroom strains, domestic and imported ogical traits. The genetic correlation coefficient of 10 hybrid oyster mushroom strains was from 0.42 to 0.80. The genetic correlation coefficients of the 4 big cup mushroom strains were quite high, ranging from 0.74 to 0.94. Through the analysis of genetic differences, author established the genetic relationship diagram of the mushroom strains. Third, the technical procedure for spawn production of big cup mushroom strain. By hybrid oyster mushroom strain, P7 with technical parameters as follows. The most suitable pure culture medium of P7 hybrid oyster mushroom strain was potatoes 200 grams plus rice bran 100 grams plus fresh mushroom 100 grams plus 0.5 grams, MgSO 4.7, H2O plus 0.5 grams, KH2PO4 plus 20 G glucose. Liter of liquid, pH 6.5. Shake rate of 120 RPM. Incubation time is 84 hours. The optimum intermediate phase spawn medium is similar to the primary propagation medium with aeration rate of 0.65 liters of air liter of medium. The optimum spawn rate used for intermediate phase culture is 10% the culture volume of substrates. The optimum pure culture medium of BI big cup mushroom strain was 100 grams bean sprouts plus 2.5 grams CNM plus 3.5 grams peptone plus 0.5 grams MgSO 4.7 H2O plus 1 gram KH2PO4 plus 20 G glucose plus 1 mg vitamin B1 liter of liquid pH 6 5 shaking speed 140 RPM. Incubation time is 96 hours. Suitable medium for big cup mushroom intermediate phase spawn was 2 grams. CNM plus 2 grams peptone plus 0.5 grams. MgSO 4.7. H2O plus 15 grams glucose plus 1.5 mg thiamine. Liters medium with aeration rate of 0.75 liters of air liter of medium. The rate of spawn used for intermediate phase is 10% of total medium. Fourth, optimization of the cultivation of P7 hybrid oyster mushroom strain and bi big cup mushroom strain by using liquid spawn on complex substrates with detail information as following. Using long-time incubated sawdust, 30-40 days, with supplement, 3 kilograms urea plus 5 kilograms superphosphate plus 15 kilograms CaCO3 plus 1 kg MgSO4, material ton. The P7 hybrid oyster mushroom strain achieved the highest yields with cultivation formula, 48% of mulch plus 48% of short-tempered bean plus 3% of rice bran plus 1% of KCO3. Optimum spawn age of 84 hours. The rate of spawn was 20 milliliters of material bag. The use of liquid spawn for mushroom cultivation can reduce cultivating cycle by 13 days. Bi big cup mushroom strain grew best on cultivation formula containing 39.5% of long incubated sawdust, 39% of cottonseed, 0.1% MnSO4 plus 0.1%, KH2PO4 plus 8% corn power plus 12%, rice bran plus 1.3%, KCO3. The optimal concentration of oxygen is 0.75 liters, 1 liter of medium minute. The rate of original spawn of grade 1 for inoculation is 25 milliliters liquid spawn bag. The most suitable liquid spawn age was 96 hours. The use of liquid spawn for mushroom cultivation can reduce 15 days cycle of cultivation. Recommendations. Basing on the obtained results, author proposes continue to research on hybrid breeding of mushrooms by using the materials obtained from results of this thesis. Applying the novel technology in propagation of spawn, cultivation on complex substrates to development of the P7 hybrid oyster strain and BI big cup mushroom strain in industry scale with high efficiency and sustainability. 25. List of publications involved in the dissertation. TT Articles Authors Publisher. Một kết quả nghiên cứu chọn tạo các chủng nằm sò mới có triển vọng trong sản xuất ở Việt Nam. Ngô Xuân Nghiên. Nguyễn Thị Bích Thùy. Trần Tam Kiệt, Trần.